Many years ago, diabetics need insulin to survive and had to inject insulin that came from cows or pigs into their body. Today, insulin used by diabetics all came from microorganisms such as E. coli and yeast. But how did these organisms produce human insulin?
Brilliant! I have absolutely no idea what's going on.
It's all thanks to genetic engineering. Genetic engineering is the process of using recombinant DNA technology for the direct manipulation of genetic materials in order to alter, repair, or enhance form or function. But the big question is, where did it all start? It is first important to learn about the evolution of genetic engineering for us to understand the current state of the field.
It all started with the discovery of DNA in 1953 by James Watson and Francis Crick. Rosalind Franklin also made a crucial contribution by using a technique called X-ray crystallography, which revealed the helical shape of the DNA molecule. Watson and Crick realized that DNA was made up of two chains of nucleotide pairs that encode the genetic information for all living things. This was one of the most important early milestones that defined genetics as we know it today.
In 1968, Werner Arber discovered special enzymes called restriction enzymes. Restriction enzymes, which recognize and cut specific DNA sequences, are used to cut open a plasmid in preparation for the insertion of a gene. The 1970s is known for the birth of recombinant DNA. In 1972, Stanley N. Cohen and Herbert W. Bauer successfully introduced rDNA into another organism artificially. The basic principles of recombinant DNA technology comprises of four major steps. A DNA fragment containing the gene of interest is obtained from a donor cell. A plasmid, commonly used as vectors, is obtained from a bacterium. They are used to transfer the gene of interest into a host cell for expression. The DNA fragment containing the gene of interest is cut using the restriction enzyme that acts like scissors. The same restriction enzyme is used to cut open the plasmid. The DNA fragment containing the gene of interest is inserted into the open plasmid through the help of DNA ligase, which acts like glue. It catalyzes the joining of the DNA fragments and the plasmid. A recombinant plasmid is finally formed and can be introduced in a host cell for different purposes like producing insulin. In 1981, the first transgenic animal was introduced. Wagner and his team transferred the gene of a rabbit into the mouse genome by using a method now standard in genetics known as DNA microinjection. Their experiments paved the way for the revolutionary transgenic animal experiments in the years to follow. In 1983, polymerase chain reaction or PCR was developed and is used to make copies of specific DNA segments. It made DNA far more accessible to scientists by reducing the time it takes to clone a DNA. In 1986, the first recombinant vaccine for human was approved. By the end of the 1980s, Bt corn, the first GMO crop, hit the fields. Known as Bt corn because it contains genes from the bacterium Bacillus thuringiensis, this corn was able to increase yields by discouraging pest impacts. The 1990s was a new period of discovery, starting with the discovery of the principles of CRISPR. Francisco Mojica discovered the principle of CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. They contain a sequence that shows up and over again, though it's often reversed each time, which makes it palindromic. With CRISPR, a group of enzymes recognize certain repeats and break the DNA there to insert important information in the middle. In 1994, genetically modified tomato was brought into the market for public consumption. It was said that this crop named as Flavor Saver, has the ability not to ripen. However, it did not meet the anticipated level of success and was then pulled out of the market. This case is a perfect example of how difficult it is to bring genetically engineered products into the market, since people are wary of the possible side effects. In 1996, humankind became witness to the first cloned mammal from an adult cell known as Dolly. Dolly was a huge achievement, proving that the process of cloning found in nature could be attributed to organisms it does not naturally occur in. Another milestone was achieved in 1999 when scientists were able to completely map out the sequence for chromosome 22, making it the first human chromosome to be sequenced.
Wow, you are almost halfway through the journey. The end of 1990s marked the progress in the mapping of the human genome, which led to further developments in the history of genetic engineering. It was a great way to open a new decade of promising discoveries and brand new experiments. In 2001, the first gene-targeted drug therapy was approved, known as Gleevec, and is used as an anti-cancer treatment drug. It works by slowing or stopping down the growth of cancer cells. In 2003, Alan Blake and Richard Crockett produced the glowfish, which is a fish that glows and became the first animal for sale as a pet that had been genetically engineered. This marked the start of an era in which the public became more open to the idea of consuming genetically modified organisms. Moreover, the World Health Organization and Food and Agricultural Organization of the United Nations developed international guidelines and standards to determine the safety of GMO foods. In 2006, a breakthrough was made in the field of stem cell research. Dr. Shinya Yamanaka introduced pluripotent stem cells technology. These iPSCs are capable of differentiating into any specific cell type that the researchers wish to investigate.
Another breakthrough in science was made in 2012 when Jennifer Duodna, Emmanuel Charpentier, and their team brought light to the biochemical mechanism of CRISPR technology. CRISPR-Cas9 technology comprises two main components, the single guide RNA and endonuclease DNA or Cas9. Here's how it works. Scientists design the single guide RNA to match the gene they want to edit and attach it to Cas9. The guide RNA directs Cas9 to the target gene and will bind a short sequence known as the PAM or protospacer adjacent motif. PAMs are short 2,6 base pair sequences in the viral genome found adjacent to sequences targeted by a Cas nuclease. Cas nucleases recognizes PAM and cannot cleave the DNA unless a PAM is present. The Cas9 will unzip the DNA and match it to its target RNA. The Cas9 RNA complex then makes a double-stranded cut in the genomic DNA, and the programmed DNA that contains the desired sequences can be inserted at the cut. This DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. What CRISPR technology is being used for varies from developing cancer treatments to tackling obesity, creating hornless cows, and so much more.
The results of CRISPR clinical trials slowly began to trickle in. Victoria Gray, who was the first patient to undergo the sickle cell disease treatment. Doctors have now treated at least 45 patients with sickle cell and a related condition known as beta thalassemia, and are reported indicating it's working for at least 22 of them. There is currently a CRISPR clinical trial for treating an eye disease, namely liver congenital amaurosis, which is the most common cause of inherited childhood blindness. Patients receive a single dose of CRISPR therapy by injecting directly into the eye. The injection contains non-pathogenic virus carrying the Cas9 protein and its guide RNA. If the treatment works, it will be the first trial leading to a direct fix for a genetic disease. CRISPR-Cas9 will also play a significant role in the future in the poultry industry by generating genetically engineered avian species with desired gene characteristics. It is predicted that the production of chickens with lean meat and lesser fat will make them more attractive for consumption.
Genetic engineering is definitely changing the game by allowing scientists to edit the genome in ways barely imaginable before. It holds a lot of new opportunities for finding treatments and is also a potential strategy for combating world hunger due to the growing human population. Genetic engineering will continue to evolve and undoubtedly continue to prove instrumental in our understanding of the natural world all around us.